So I think it would be most apt to ask Dr. Tatenda Dale to please start off this session and give us an overview on the project, and then we can move on to the rest of the presentations. So Tatenda, if you would be so kind.
Okay, good afternoon, everyone. For this session, as most of you have sort of like realized, is that most of the presentations are based on the Kakea Bray transboundary aquifer region. So it is a project that is funded through the SASAqS GMI, and it's funded by the JRS Biodiversity Institute. So the project involves trying to understand the biodiversity that exists within these transboundary groundwater dependent ecosystems. The project is sort of like three main components. That is the biodiversity, where I'm the biodiversity leader working with Professor Wasserman as a co-leader for that section. Then we have the hydrogeology that is led by Prof. Gomo at the University of the Free State. And then we have the GIS and remote sensing led by Farai and Prof. Dube. All right, so all these different sections, they sort of like work together, they are integrated, and we want to find how best these groundwater dependent ecosystems sort of like integrate, and how they function and how they depend on the groundwater. So the remote sensing will come in to try and sort of like spatially map the species diversity that exists within these environments. So the study is being conducted within South Africa, in the Northwest, and also in Botswana. So it's a transboundary study that is mostly focusing on groundwater dependent ecosystems within the Kakea Bray dolomite system. As some of you might not know, the Kakea Bray transboundary aquifer system is a huge system that covers South Africa and Botswana, but the specific groundwater dependent ecosystems that we're mostly interested in are mostly located within the dolomite region of the Kakea transboundary system. So it's a narrow range that stretches from Morokweng, from the Morokweng area in South Africa all the way to Botswana, sort of like the southern part of Botswana. So in Botswana, what we have is sort of like a small section of the dolomite range where we are able to find a groundwater dependent system. So the students that are going to present their projects will sort of like try to highlight some of the preliminary findings that they have, that they currently have, and I hope you will find their presentations are interesting. Thank you.
Great, thank you very much, Tatenda. This is a very nice big project that you've got going here, so I'm looking forward to just hearing a bit more from the students and the other co-workers. So with that, I would please like to ask Mangana Ramferi to tell us a bit more about the progress that has been made regarding groundwater dependent ecosystems. If you're there. Mangana, are you there?
Hello.
Hi.
Okay, thank you. My name is Mangana I'm from the University of the Western Cape. So our work is based on literature review, where we are looking at the progress in the remote sensing of groundwater dependent ecosystem in arid and semi-arid environments. So I will just go straight to our findings. So we found that there is a progress in the use of remote sensing in GDE. So the study found that 2080 is the year with the most published article or scientific papers in the use of remote sensing in GDE. So from 2019 to 2020, there was a decrease in the publication. So, however, from 2020 to 2021, there's an increase in the publication. So, we also investigated the remotely sensed method used in this GDE study. So, we found that the most used satellite images were Landsat and Sentinel datasets. So, in terms of machine learning algorithms, we are having indices and image classification. So normalized difference vegetation index, in short NDVI, and normalized difference water index, in short NDWI, were the most commonly used indices. And in terms of image classification, supervised classification is the most commonly used classification. So this study found that the used method are somehow associated with some challenges or pitfalls when used individually. So this study recommends the integration of all the mentioned method, and also integrate with other remote sense method data like digital elevation model and climate variable to improve the result produced in the use of remote sense data in GDEs. Thank you.
Cool. Thank you very much, Mangana. Next, I would like for us to keep with the same topic and then just ask, with the same topic of remote sensing, so I would like to ask Kudzai Mpakairi to tell us about the use of remote sensing to identify conservation areas.
Good afternoon, Lizanne.
Yes, that's me.
I hope I got that one correct. Sorry.
Yes.
Yeah, my name is Kudzai Mpakairi from the University of the Western Cape, and my presentation or my talk, which is already on YouTube, focuses on mainly identifying priority conservation areas within groundwater dependent ecosystems, specifically focusing on the Kakabray transboundary aquifer. So in this study, mainly we focus on using remote sensing data to calculate species diversity. And specifically, we calculated the species diversity using a new measure called the SASAqS. It has been used before in looking at the phylogenetic diversity, but in this case, we used it with remote sensing data under the presumption that spectral variability is related to ecosystem heterogeneity, and from that we can calculate species diversity, and then from the species diversity that is calculated or that we can calculate, we can then use that to look at the ecosystem health or integrity, because the assumption is that healthy ecosystems are more diverse as compared to ecosystems that are less healthy. So then once we calculated that the SASAqS, the species diversity from the remote sensing data, we validated it with field measured data on species diversity, and we were capable of then pinpointing where the species diversity is within the Kakabray transboundary aquifer and identifying priority conservation areas within the Kakabray transboundary aquifer. For example, we observed that species diversity was high along the fence lines, was high along the roads, around natural water pans and rivers. And in this case, for example, around natural water pans and rivers, this is not surprising because these systems are also supplemented by groundwater. So it would be expected that the species diversity around those areas would be high because groundwater is available to these ecosystems. And understanding that the Kakabray is an arid environment, the availability of groundwater then allows for the existence of vegetation or the proliferation of vegetation even during the dry period. So that was the focus of this talk, in trying then to use remote sensing to identify the priority conservation areas. So you agree with me that the focus of the study was just not to identify the priority conservation areas, but it was more focused on where do we need to put the effort when it comes to conserving priority conservation areas, because you agree with me again that effort is important, but knowing where to put the effort is what makes the difference. For now that we can do that, we can even scale this from the Kakabray to even the whole of South Africa in identifying priority conservation areas that need to be monitored and where we need to channel our resources towards. So that was the main focus of this talk and presentation. Thank you very much.
Cool, thank you very much, Kudzai. Now, after we've decided which where to put our priorities and efforts in terms of conservation, I think we move into biodiversity of the ecosystems in this transboundary aquifer area. So I would like to invite Tafara Bute to please tell us about the microbial biodiversity of these ecosystems.
Okay, hello everyone, can you hear me?
Yes, we can hear you, thank you.
Okay, yeah, my name is Tafara Bute, and I'm a master's student in the Department of Zoology and Entomology at Rhodes University. And my work is still going on, and I'm still busy with the lab work, but I'm gonna give you the details of how I'm carrying out my research. So the background of the study generally is that there is generally overexploitation of groundwater from the Kakabray transboundary aquifer, which has been mainly used for agriculture and domestic use. And this has greatly reduced the amount of water available to support biodiversity in the region. And this has some implications for primary productivity dynamics. So the motivation of my study is that there is no knowledge on how loss of microbial diversity will impact ecosystem functioning. Therefore, this study is aiming to conduct a baseline ecological assessment of microbial diversity of the freshwater ponds and evaluate their role in the diet of major secondary producers and also contribution to extra polymeric substance production. We are talking of carbohydrate and protein. So sediment cores will be collected from 20 ponds within our study area, and samples will be collected along a transect from the center to the edge. And this study will be carried in the wet season and dry season. So for analysis, Illumina-MISEQ high-throughput sequencing will be used to analyze using meta-barcoding approaches. That is, we are targeting the 16S RNA gene for bacteria and 23S RNA and ITS genes for the eukaryotes. To determine extracellular polymeric substance production associated with microbial activities, carbohydrate concentration in sediment samples will be measured using the phenol sulfuric acid assay by Underwood, and total protein analysis will also be conducted using the modified Lorry procedure. So the expected outcomes from this is generally microbial patterns are expected to differ within and across the testing ponds, with a higher diversity expected in wet season compared to dry season. The same also for extra polymeric substance. So the results from this study will generally provide an insight into microbial dynamics and associated extracellular polymeric substance production in relation to productivity dynamics, with implications for change in climatic conditions and land use in the region. That is generally what I am focusing on. Thank you.
Great, thank you very much, Tafara. I'm looking forward to finding out what what interesting results you get from this project.
Okay, thanks.
Thanks. So next, moving on to slightly larger organisms, I would like to invite Chipo Mungenge to tell us about the plankton diversity in this transboundary area, please.
Thank you so much. Good afternoon. My name is Chipo Mungenge, and Biodiversity. I'm part of the Wetland Ecology Lab under the supervision of Macroinvertebrates. And as already explained by Dr. Dalu, this is my study is part in the SASAqS transboundary aquifer pond system by the SASAqS GMI Institute. The reason why I'm carrying out the study is because currently there's very little information on plankton fauna in temperate wetlands, and therefore we will be looking at the plankton diversity in the pond systems in the SASAqS transboundary aquifer. We'll also be looking at the food web structures in the ponds through stable isotope analysis, and also with salinization being recognized as a major threat in most freshwater systems, which is being caused by anthropogenic activities and climatic processes. We will also be carrying out lab experiments to determine whether salinity affects hatching success and germination success of plankton from sediments that were collected from the basins in the dry season in June. The expected output of the study is to create a database on plankton species in the pond systems. Thank you.
Great, thank you very much for that overview. I wish you all the success for this project. Finally, moving on to even larger individual or organisms, I would please like to ask Fanny to tell us a bit more about the macroinvertebrates of these aquifer systems, please.
Afternoon, everyone. My name is Fanny Masina. I'm a student at the University of Mpumalanga, the School of Biology and Environmental Science. So my study aimed to investigate habitat quality using microinvertebrate community in the SASAqS transboundary aquifer region, as this region is threatened with degradation, and also this region is completely understudied from a wetland perspective. As such, ecosystem services stability and strength to those wetland are poor understood. So with that, specifically, we are comparing the diversity and community composition of microinvertebrates with the focus on the pen size and also the level of disturbance. So in my study, three objectives. Each one we're going to identify the major disturbance to pen system. Objective 2, so we will assessing the water and sediment chemistry in relationship to the pen size and disturbance. But the objective 3, where we determining the relationship between microinvertebrate diversity and disturbance. So far, we have went to collect data out of first sampling, and then we collected microinvertebrate on different wetland pen system, sedimentary sediment, and also water samples. And the data is still in progress. We are still analyzing the data, and the results are quite interesting so far. Also, we are expected to go to the field maybe next month to do our next sampling. After the study have been completed, the study can help to prevent further loss of wetlands in this region by implementing conventional conservation plannings. Thank you.
Cool. Thank you very much for that. I hope your fieldwork next month goes really well and that you find some interesting results. With that, the presenters have given us a bit of an overview of their work, and now I invite everyone to the question and answer session now. So if you have any particular questions, please put them in the chat box or raise your hand, and then you can ask the questions to our presenters.
Okay, we have one question here for Kudzai from YouTube. Someone said it's a great presentation and a really interesting work. What he did comment on and ask maybe a question was, he said it would be good for future studies to reflect on strategies to remove the effects of invasive species on the resultant map, as it may give misleading impressions, particularly when GDE maps are to be used to locate conservation areas. Your observation that such phenomenon potentially occur close to roads would be a good starting point. Not sure if you want to comment on the invasive species aspects regarding remote sensing.
Thank you, Wynand, for that comment. Yeah, the concept was that because along your major roads and along most roads and within this environment, the Kakebwe, you'd realize that the movement of vehicles or the constant disturbance in trying to maintain these roads, they make the roads susceptible to invasion by invasive species. So identifying these conservation areas now, or identifying these areas as with high species diversity, it also helps, for example, the likes of SANBI to focus on because understanding that these areas of high species diversity is important, but now we are more or less moving towards that. What is the reason behind this species diversity along this road, along the roads? So in this case, like for the Kakebwe, like from our observations, we know that along those roads, it's more likely that that high species diversity is more related to invasive species, and then it means that we can focus, or in terms of looking towards eradicating or removing invasive species within the Kakebwe, we can more or less focus along roads, and then, but definitely going forward, if we are to only look at species that are important to the groundwater dependent ecosystems of the Kakebwe that are not invasive or anything, it would be very important. And I think it's something that I would take note of. It would be important to take out and eliminate those areas, and then focus on calculating the species diversity with those areas removed from the map.
Okay, thanks. So just a follow-up, maybe more out of ignorance. So can you identify, if you don't look at the roads, so there's no roads, can you identify invasive species with remote sensing, or is it not fine scale enough? It's more out of ignorance, I'm not really sure. That's what I'm wondering.
No. Yes, the assumption with remote sensing is that each and every species on Earth is a unique spectral signature that we can identify from space or in the air. So because each and every species has a unique spectral signature, it means identifying it is easy. What we then have to use to identify that species is something else. That's a whole different issue in terms of the sensors and the technology that we need to use. But it's very possible for us to actually identify invasive species within any environment with remote sensing.
Cool, thank you very much for that. A follow-up question just in terms of that, also out of ignorance, do you have any idea which invasive species occur there? Is it like mostly terrestrial, like trees or so on? Do you have any idea what invasive species they are?
Thank you for that one. I forgot. I think I did not mention this one when I was giving this more talk. For the species diversity, we focus mainly on vegetation diversity and not faunal diversity. So it was mainly floral diversity. So most of the species that we found were your typical grass species that were there, but these are not the invasive ones. Some of the other species that we also identified that we assume might be invasive, we took them to the lab. So we're still waiting for the botanists to identify these species for us and tell us if they are invasive or not, because we could not find them in any literature or vegetation literature within the Kakea Bay. So our assumption is that these species are more likely to be invasive. But yeah, that's where we are now.
Great, thank you very much. There's a comment from Matthew Bird in the comment section that just says, great project, guys. Nice to see groundwater linked systems getting some attention in the freshwater ecology realm. Also very nice to see the transdisciplinary aspect that this project involves. And I do agree. I think it's really nice to have this kind of transdisciplinary broad project on such an ecosystem. Are there any other particular questions? Bainant has another question.
Hi, I think this question is probably more for Tatenda. I know I maybe missed it in the presentation, the length, first of all, the length of the project, and sort of idea if you're going to sample the complete whole length of time of a project at this system, or is it only going to be 1 or 2 collections for these students, master's projects or PhDs, I'm not sure. And then what is the outcome at the end? Is this going to be published on some kind of database? What is the sort of, hope for the project and the results that this project will generate?
Okay, yeah, thank you for your question. I think my presentation didn't cover like the other aspects because I focus mostly on the biodiversity within these transboundary ecosystems. But the other aspects, especially if you look at the presentation by Kudzai and Mangana, the way that they come into the project and link up with the biodiversity group is that at the end, towards the end, when we have all the different species list of the different taxa that occur within these transboundary pools, the GIS guys will come and do a spatial sort of like analysis to try and map out where the species will likely to occur based on the data that we'll have collected. And then the link to the hydrology team, they are trying to tell us how sort of like the frequency of the hydro periods within these groundwater ponds, and how often does the groundwater sort of like fluctuates. And if you look at these students, they're going to work in both countries, South Africa and Botswana, and I think it would be sort of like interesting for them based on the species from a species biodiversity assessment, and also the links between with the other two components is to provide sort of like interesting findings, because as we move along the Kakea Bray transboundary aquifer, particularly along the Dolomite zone, it sort of like disappears as you move towards the Molopo River. So there's a huge concentration of the groundwater dependent ponds around the Morokweng area. And then there's another smaller sort of like concentration of these groundwater dependent systems in Botswana. So it would be a nice sort of like a comparison across sort of like larger geographic scales. And also, we are not going to sample all the pores. As you saw, like from the video in my presentation, there's so many of them. We're just what we did when we went to the Northwest, we sort of like randomly selected a few ponds that we think might be representative, but and we also hope in future we'll be able to just go and focus sort of like on the Northwest section and do a full study where we sort of like try and assess other research questions that we are currently not addressing in the current project. Thank you.
No, thanks for that. Yeah, I was actually wanting to comment on some of those pictures in your video where you showed all the almost, it seemed like hundreds of different small pans. It's really impressive and pretty cool. And to echo what Matthew said, it's amazing and really good that these groundwater linked ecosystems are getting some attention. And just in terms of the hydrology that you mentioned, maybe you know, I mean, I don't know, that's why I'm wondering, does some of these systems remain perennial, or do they dry up, rainfall dependent, linked to the groundwater, or is it sort of some of these plants always have water?
Okay, yeah, that's an interesting question. Yeah, that's a very good question that you asked. A few of these systems, depending on how good the rains are, will have water throughout the year, but a number of them, they dry out, especially during the winter, moving to the sort of like the hot, dry months, September, October, and they sort of like start filling up in November, December with the first rains. So what we expect, especially the groundwater team, to tell us, because there we know that the groundwater levels are high, and now one of the questions that the hydrogeo team needs to find out what is the frequency of that discharge from the groundwater to these groundwater dependent systems. And if they can tell us that, we can be able to sort of like predict sort of like the emergence of some of the taxa that exist within these systems. So we know that it's a combination of groundwater and also surface water that fills these systems, and the groundwater sort of like plays a role in maintaining the water levels within these systems. But there are challenges that might affect these systems, such as the intensive agriculture that is now taking place in some of the especially within the commercial area, where now they are moving into sort of like this huge potato farming, which requires a lot of water. So they are doing a lot of groundwater extraction. So we might start seeing shifts in the length of the hydro period for some of these groundwater systems. But we still have to investigate that for us to conclusively say that the water extractions will impact on these systems.
No, thanks for that. Really interesting. Looking forward to the results coming out of it.
Thank you, Winners.
All right. Do we have any other questions or comments from the audience? I just had one more question for Chipa. I just wanted to know, I saw that in your presentation you said that you'll be doing the hatching at 18 degrees Celsius. Now I just wondered why such a cool temperature, because from what I know in the pan systems is generally the water is quite warm and the hatching is also generally at a little bit of a warmer temperature. So I was just wondering, do you have any specific reason for doing the hatching at 18 degrees?
Yeah. Yeah, I heard it.
You're very quiet, Chipo. Could you just turn your volume up a bit, please?
All right, let me try to. Can you hear me now?
Yes, that's much better. Thanks.
All right, sorry for that. The 18 degrees was just the temperature of the fluorescent lighting in the rooms that we'll be using.
Oh, okay. All right, thank you.
Can I come in there? I think one of the other main reason why the temperature was selected is based on some of the work that Ryan did in Botswana. When he was still in Botswana, I think they managed to find that temperature, they managed to sort of like hedge a lot of stuff. And yeah, for Chipo, yeah, I know he she wrote 18 degrees, but we expect to play with the range of different temperatures to see which temperature will be the best, because maybe 18 degrees won't work for those systems.
Okay, cool. Thank you. Thank you for that. I don't know if there are any other burning questions from the audience or any comments. Otherwise, we'll end the session there and take a quick. Oh, sorry. I see there's a hand from Richard. Yes, Richard, you can ask your question.
Thanks, Luzon. Sorry, Tatenda, I might have missed it, but have you thought about doing eDNA to look at the different diversity that's available?
Thank you, Richard. Yes, yeah. So the MSc student Frank and also Chipo, Ryan indicated that since we won't be able to go as frequent as we would like, so the best other alternative was to do eDNA. So we've been working with Adam, who's an aquatic microbiologist, to try and assist us getting the eDNA component going, and we hope that at least we'll try to get a more comprehensive species list based on that, and also there are other components of the study, especially if you look at the vertebrates, which were not presented at this conference, which are currently being led by Chad, who's looking at the frogs and also the different reptiles that play around within these pores. So we hope that when the project completes in the next 2 years, we'll have a full sort of like a comprehensive list based on sort of like the normal taxonomic analysis, and also from the eDNA for the invertebrates and vertebrate groups. Thank you.
All right. Vainant, I see you have your hand up. Would you like to ask a question?
Yeah, it's probably more of a comment to Richard, and I think the problem with the eDNA and a lot of these wetland systems for the plankton and but there's not always reference or barcodes available for these invertebrates, being it plankton or macroinvertebrates, especially down to species level, which makes the eDNA useful to some degree, because you'll get operating sort of taxonomic units, but not very specific for the biodiversity. So, yeah.
Yes. I totally agree with Feinat, because that is one challenge that we discussed with Ryan with regards to sort of like getting whether eDNA will provide a comprehensive sort of like the species list. We know that, yeah, we're going to do the eDNA, but we won't get a comprehensive list because most of the taxa that are found within these systems are not yet listed on whatever GenBank or some other repositories that exist for that component. Thanks.
Cool. Thank you, Wynand and Tatenda. I see, Ryan, you have your hand up if you would like to weigh in or make a question or comment.
Thanks. Thanks very much. Yes, the nice thing about collecting the samples in the way that we're doing, or Frank in particular is doing, is collecting sediment samples and water quality samples, and is extracting the DNA. And then once you've got to that point and everything's extracted, you can start looking at different things. One of the things we were talking about kind of exploring for the eDNA approach is the large brachiopods, where we could easily develop kind of a reference collection and then develop or see if the eDNA approach will work for that, and then try and build up on that. But you are going in blind. We can get really nice alpha diversity lists or OTUs, but other than that, it is limited. It has a kind of limited application, but it is something we're thinking about, and we are collecting the samples, so we'll be able to explore options. Thank you.
Yeah, no, I agree, Brian. I mean, I was also wondering about the large brachiopods, because it's probably almost the most available reference list could probably be found in the large brachiopods in the system, and especially if you could isolate it from the sediments, so you don't have to worry about it being actually there. It's raining and it's no water. So yeah, it's good. Nice to see this sort of projects coming about and starting to get some traction.
Tatenda, your hand was up, but I don't see it anymore. Did you want to make any final comments?
No. I think I just wanted to highlight and support what Wynand was saying. Thank you.
Okay, cool. Thank you very much. All right. That was a very nice discussion that we had there at the end. I hope everyone enjoyed this session and the presentations. And please, if you haven't watched any of the presentations yet, please do go do that. They are really, really nice. And leave any comments or questions or just a like. And the poster session will start at 3. But Wynand just wants to say some things. I just wanted to finally close this session by saying thank you to everyone for participating. Thank you to all the presenters. Thank you for everyone who asked questions. And I will hand over to Wynand now.
Thanks, Lizanne. So just before, if you want to take a quick comfort break, in the break, maybe just everyone be back just before 3. Don't have to necessarily take a long break. I'm going to play all the videos of the posters here so you can see it, in case you haven't had a chance to go and watch it. And then we'll start at 3 o'clock or just before 3 maybe with the posters if everyone is in attendance. So thank you, and I'll see you in a few minutes.